a very relevant subject for modern agriculture. And to chair that, I welcome on stage two professors from our School of Biotechnology. The session will be chaired by our senior professor, Professor T.K. Adhya. Sir, please come on stage. And the session will be co-chaired by Emeritus Professor, Professor K. Jena. Sir, please come here. So Professor T.K. Adhya has a stellar career. He started his academic and research career back in 1973. He has served as the director of Central Rice Research Institute, CRRI Cuttack, in 2011, and he joined School of Biotechnology, KIIT, in 2012. He is also seconding as the director of South Asia Nitrogen Center, New Delhi, since 2016. He has been part of several prestigious national and international collaborations, and also have been part of different committees of the government and policy making related to agriculture and use of modern agricultural practices. He has over 200 high impact publications, and he has been a loving colleague guiding us all at the School of Biotechnology since we have been working here. And so Professor Adhya, you can now start chairing the session. Yeah, sorry, speaker.
I think I can start it as a good afternoon. We are already late by 45 minutes. And I think we have to really tighten our belt, because after that we have to loosen it for lunch. So for that, the time we will try to reduce a little bit. We had 2 lectures in the pre-lunch session and 2 more in the post-lunch session. I will, rather with the help of my colleague, Dr. Jena, a learned international level scientist on rice, we will be working, we will be trying to manage the whole session, probably within time, I'm not sure. But since lunch is there, I think we will be trying to push it a little faster, because for that only we live. And then we have to manage that. So the next, the session is actually the modern technology and biotic resistance. So I do not want to do a preparatory talk here, but rather introduce my colleague speakers who will be speaking, and they are all learned people. Probably I can learn many of the things from them. The first speaker is Professor K. Jena, K. Jena rather. Professor Emeritus, School of Biotechnology, KIIT University. He had an excellent 35 years of research and teaching experience, and completed his PhD from Utkal University. In fact, he did his PhD from my institute where I was there. That time it used to be called a Central Rice Research Institute, under a learned cytogeneticist, Dr. Misra, who used to, with him he has worked. And then he did his postdoctoral research at the International Rice Research Institute, Philippines, and developed a set of primary trisomics in rice, and successfully transferred different biotic stress resistance genes from 5 distantly related wild Oryza species. Then he continued his work in IRRI, and then he also subsequently went to Korea, South Korea, did his work for quite some time there. And now he has come back to his motherland and joined us. We hope that his lecture will be quite attractive, and he will be speaking on exotic genes from Oryza species, confer, broad spectrum disease and insect resistance of rice. Dr. Jena, please.
Thank you, Dr. Adhya. Good morning to all the committee members of this 5F ATPBR conference committee. Good morning to all my faculty colleagues. Good morning to all the brilliant students of our School of Biotechnology, as well as from different national universities who have come here to listen the most essential points from this conference. First of all, I am very grateful to the founder of 5F ATPBR Foundation Chair, Dr. K. Narayan, for suggesting me to give a talk on this particular topic, exotic genes from Oryza species confer broad spectrum disease and insect resistance of rice, which is very relevant to the theme of this conference, realigning Indian agriculture for a sustainable future. So I'll be presenting on this particular topic about the work that I have done in my laboratory in the International Rice Research Institute and how we have collaborated to improve disease and insect resistance in different rice growing countries in the world. It is not moving. Next slide. So my presentation will be covering, I'll be giving some introduction about this particular topic. Then I will explain about biotic stress resistant genes and what should be the research strategy at the current stage of this cutting edge science of biotechnology, potential resistant genes and their functions, transfer and expression of selected resistance genes into elite rice cultivars for sustainable production. And then I will give some conclusions about the talk. In the introduction, I would like to say that, which was published a decade ago in the magazine Economist, which was predicted that by 2050, world population will be increasing enormously. And it is predicted that it will be 9 billion population, particularly in the developing country, where the people, they live on consuming rice. And those countries are South and Southeast Asian countries. So this topic is very relevant to the audience here, that we should work how to feed the population, which feeds half of the world population by consuming rice. So it is the rice dependent population that will double by 2050, as I mentioned. And also, in spite of this, we have to develop new rice varieties 2 or 3 times from the current yield potential. But how it will be possible if you do conventional breeding technology? It needs the cutting edge science and biotechnology. Nevertheless, we have to do it in spite of having less water, less fertilizer, less fungicide, and less pesticide, and limited land. So we need to improve rice production. Next slide. So in this context, our goal is to improve genetic resistance potential of elite indica rice varieties. Why I mentioned here indica rice varieties? Because we all eat indica rice varieties. We all eat indica rice, not like Japan and other countries who eats Japonica rice. So the hypothesis here is to identify resistance enhancing functional genes controlling pathogenicity and insect feeding habits that can be introduced into elite indica rice varieties and pyrimidine resistant genes to develop durable resistance against pathogen and insects, and hence producing sustainable yield of rice varieties. Next slide. So you have to understand what are the exotic species. So for that purpose, I put this slide because the rice that we eat belongs to the genus Oryza. It is a monocotyledonous crop. It belongs to genus Oryza. It has 24 species. But the rice we eat, that is the cultivated rice, which is the Oryza sativa, which is in the form of cultivation. But there are other 23 species. Of course, there is another African cultivated rice that is Glaborima, but there are 22 species which are exotic. They appear in the form of wild morphological features. That's why I have classified those species into 3 different gene pools. One is the primary gene pool, secondary gene pool, and tertiary gene pool. And it is enormous difficulty to transfer the genes from those wild species or exotic species of the genus Oryza. But it is possible how to transfer those genes, because those wild species or exotic species of genus Oryza, they have high degree of potential for a disease resistance, insect resistance, nutrition quality improvement, as well as increasing yield potential. So I mentioned here that in my talk, I will be mentioning about disease and insect resistance potential of those wild species or exotic species of the genus Oryza. Next slide. So in this context, my talk will be covering 3 different biotic stresses. One is the disease stress, which is having 2 major diseases. One is the bacterial leaf blight caused by the Xanthomonas oryzae, Pathovar oryzae, or XOO. And the second major disease is a blast disease. It is a fungal disease. It is commonly called as pericularia disease. And it is a fungal disease, and then that is also causing severe damage. As you see in the picture, bacterial blight damage occurring in the field, blast disease damage occurring in the field, and also the third one is the insect damage occurring in the field, which is the brown plant hopper. In this context, I want to mention here that BPH damage is causing serious problem in the western part of Odisha for the last 3 or 4 years. In the western districts of Oryza, the rice crop completely damaged by this BPH infestation in the field. And the field appears like that. So the farmers, they lose sometimes complete loss of the crop. Similarly, in case of bacterial blight, in case of severity, there will be about 20 to 80% yield loss will be there. But because of the advances in biotechnology, there are about 42 bacterial blight resistant genes have been identified. And out of those 42, 9 resistant genes are cloned and characterized. Among the fungal blast resistant genes, in the extreme cases, there is complete loss of yield. Through research, about 100 resistant genes are identified, and out of that, about 20 genes are already cloned using the cutting edge biotechnology. Among the insect damage caused by brown plant hopper, it is a sap-sucking insect, and then it is causing serious problem of hopper borne, as you see in the top picture, how it is happening in the rice field, and then there is a complete yield loss. In addition to that, that particular insect is so damaging that it transmits 2 different viral diseases. Then the rice crop gets stunted and will never flower. In spite of that, because of scientific efforts using mostly genetics and biotechnology tools, we have identified 33 BPH resistant genes and incorporated into different rice varieties for protection. And using biotechnology, we have already cloned 8 resistant genes for brown plant hopper. Next slide. So coming to the first one, which is bacterial leaf blight. It is caused by Xanthomonas oryzae, Pathovirus oryzae. Yield loss is very high. So the best thing is the host plant resistance by conventional technology, which is going on for several decades. But with the advances in biotechnology, we are now trying to use the cloned genes with a known function for resistance. Earlier, we were doing only identifying the gene and transferring by using the conventional genetics method. But now using the biotechnology, we have to understand what the function of that particular gene so that the pathogen will not attack the plant. So in that context, we have done some research on the wild species by evaluation and knowing their potential for resistance to bacterial blight disease. And there are about 10 resistant genes from the wild or exotic species of Oryza were identified in different laboratories in the world, including my laboratory. And then in that, 3 genes are already cloned and characterized. Their functions are known. So if in this 21st century, while approaching to build up resistance in any crop plant, our students must know what is the function of that gene, whether that gene can restrict the invasion of that particular pathogen into the rice vascular tissue. And it is possible. I'll show you some examples. Next slide. So here I have listed about several important resistant genes of bacterial leaf blight. Some of these genes are derived from the cultivated rice varieties or land races. But my talk is about exotic species used for genes. Among that, one gene is XA21. That gene is isolated from Oryza longistaminata, which is one African wild species. Oryza longistaminata. You see, after cloning that gene, it is clearly understood that that gene encodes a serine threonine kinase protein. That serine threonine kinase protein, which is a receptor protein, which will be interacting with the effector protein of the pathogen and gives very strong resistance to the invasion of the pathogen and makes the rice plant highly resistant. And that work had been done in University of California, Davis by Professor Pam Ronald and his students. Many young students, they worked to clone that particular gene. Similarly, the other gene from Oryza minuta from XA27, and they are located on different chromosomes. And the gene I have identified is the XA40 gene, which is encoding wall-associated receptor kinase protein. So that we call it WAG3 gene. That WAG3 gene is from Oryza australiensis. And these 3 genes are very important. They are from the exotic species of the genus Oryza. Next slide, please. Here I give one example of that identification of XA40 gene from the exotic species Oryza australiensis. You can see in the left side how that leaf, because this bacterial blight is a vascular pathogen. It invades through the vascular tissue. And you can see after infection, that pathogen cannot invade the vascular tissue and the plant remains resistant. But the rice varieties without that gene, that is showing high susceptibility, as you see here, the lesions, how it is spreading in the leaf. So it is that's why it is leaf blight disease. Okay, so this gene XA40 is a broad spectrum bacterial blight resistant gene and has a high genetic combining ability. That is most interesting part, that it can be combined with any indica rice varieties. Next slide. So here I am giving you the evaluation potential of this particular resistant gene XA40. We conducted experiments by isolating different pathogen isolates in Philippines, including most highly virulent pathogens, which are named as PXO pathogen, Philippine Xanthomonas pathogen. You can see that how it is showing all the strains of that pathogen. It is RRRR like that. It is highly resistant. Whereas there are the rice varieties showing high susceptibility at the top two lines, which are from the cultivated rice varieties. In addition to that, through our screening, we identified several other species. They have some future potential to use it for rice improvement program from different genomes of genus Oryza. Next slide. Here, I am giving one example of another species, which is the Oryza rufipogon. Oryza rufipogon is a wild species, which is the progenitor of our cultivated rice. But nobody has explored its genetic potential, whether it can have some potential genes or not. So at IRRI, what we did, we screened several accessions of Oryza rufipogon. Then we found that one particular accession is showing very broad spectrum resistance against many virulent strains of that pathogen. So that we picked up that particular species and then conducted genetic analysis and then molecular analysis to identify the gene for its future integration into different indica rice varieties. Next slide. Here I am giving a comparative picture of earlier identified gene and the present one, which is from rufipogon. As you see, I have given the comparison of different virulent strains, res 4, res 6, res 10, and I compared with the XA21 gene, which was earlier cloned and characterized, but because of the climate change and other effects, the pathogen also changing their pathogenicity. So that gene is getting moderate resistance to one strain, but the new gene from Oryza rufipogon is giving consistently very high level of resistance to all the virulent strains. Next slide, please. So we continue to identify what is that gene present in that Oryza rufipogon. And as you see, we identified two genes for XA45 and XA46. And you see the phenotype picture by after infection to the leaf, it is giving very strong resistance that it is not having any spread of infection to the leaf. Next slide, please. Then coming to the second most important disease occurring in the rice plant in the field is the blast disease. So blast disease is caused by the fungus, which is the Magnaporthe oryzae or Pericularia oryzae. So this Pericularia oryzae is a very serious disease in different parts of the rice growing countries in the world. So far, 100 genes are genetically identified, but by using biotechnology, we have now 18 genes which are cloned and characterized. So once you characterize, you will be knowing very well what the function of that particular gene you have cloned. Then you can safely use it for transferring that gene into different susceptible rice varieties. So because of frequent breakdown of rice, but this Pericularia disease is having very complex genetic mechanism. There is very quick breakdown of resistance. If some genes are introduced into the rice variety, you need a durable type of resistance which can last for longer period. So we have identified the resistant gene, and also we have done pyramiding of different resistant gene so that it can have better durability in the rice varieties where it is getting introduced. Next slide, please. Here I have given one model that how you will be quickly evaluating the magnitude of resistance by putting the gene into the rice varieties. So this is called sequential planting method model, and that work has been published in Theoretical Applied Genetics and Phytopathology journals. And as you see here, that the lines that we have developed, that particular gene from Oryza australiensis is PI40. So Pericularia and number is 40, that gene. When we identified and characterized that gene, it is published in the journal Theoretical and Applied Genetics. And there are several lines I have given ABL 1, 2, 3, 4. So you can see the degree of resistance is very strong as it is done in a in the screen house with a genetic model. We have made the sequential planting method, which is a model that you can collect the virulent strains from different parts of the field, where there is a hotspot for that fungal disease and mix it, and then you can conduct the experiment in the glass house by giving sequential seeding of the rice varieties and expose to that particular fungus, and then see how it is durable, how many cycles it can sustain its resistance. So that is a picture you can see. It is giving no susceptibility at all. Next slide, please. So the earlier conventionally identified genes, as you see here in the top in this graph, they are showing high degree of susceptibility because of the change in the pathogenicity of this particular fungus. But the new gene, that PI40 we have identified, is in the lower side. It is giving consistently resistant in all the 7 cycles that we have exposed the plants to that particular pathogen. So it gives us the information that PI40 gene is a durable resistant gene in different strains of this Pericularia fungus. Next slide, please. So as some students are here, so I put this picture how you can monitor that, collecting the rice plant, extracting the DNA, and then you can use the gene-specific DNA markers, and you can analyze. If you get the banding pattern, then you will be able to see the presence or absence of the gene. Then you will be conferring that this particular variety is going to have high degree of resistance against this particular pathogen, which is a fungal pathogen. And then you will be recommending to the farmers to grow this particular variety, even though there will be future attack of this disease in the field. Next slide. So we evaluated that PI40 gene in the greenhouse by exposing to the most virulent strains. And you can see here the picture that the lines that we have developed, they were exposed, but there was no infection at all, compared to Co39, which is highly susceptible. You can see the disease lesions there in the left leaf. And also in the screening picture, you can see Co39 is a standard check variety for high susceptibility to blast disease. But the traditional variety, which is from the cultivated germplasm, is also showing very high susceptibility in the right side. But in between, you can see the gene PI40 is showing all the green leaves. There is no infection, and it is giving very high level of resistance to the disease. So that is the outcome of this biotechnology, that you can change the susceptible plant to resistant by incorporating the resistant gene, which is identified by using the cutting edge biotechnology techniques. Next slide, please. Then I was mentioning that to make the plant having durable resistance, because you never know that because of the climate change and other effects, there is breakdown of resistance. So in your hand, by using the biotechnology tools, you can do pyramiding of the resistant gene, and then you can see the picture how the pyramidal lines are showing very high level of resistance. So they are not at all getting infection by the virulent strains, and you can evaluate and know that it is having high potential for a resistance. Next slide. And this is also again evaluation picture, and side by side you can check when it is evaluated and showing resistance. Then you can check the DNA profile, and you can see that the gene specific marker bands are present there or not, and it is present there. That's why it is causing high level of resistance. Next slide. And here the question is that, can you do the pyramiding of bacterial blight resistant gene and insect resistant gene and also blast resistant gene? We have done that, and you can see in the last row, it is showing complete resistance to blast, bacterial blight, and also BPH. So it is called pyramiding effect to have durable type of resistance. Next slide. So we conducted the experiment at the International Rice Research Institute, but our main purpose is how to help the national program to utilize these lines so that they can have better resistance to the local rice varieties. So we collaborated with the National Rice Research Institute in Cuttack and also Indian Rice Research Institute in Hyderabad. So they found that some of these exotic gene pool lines, they are showing high level of resistance. The green lines, you can see resistance against the blast pathogen. And that has been showing very high level of resistance to also bacterial blight pathogen in our country. So now the scientists in those two institutes, they are now identifying what is the gene present there using the tools of biotechnology. Next slide, please. Next slide. Okay, this is the experiment the scientists in these two institutes they have conducted, and they have found that these lines are having very high level of resistance. The genes are derived from the exotic gene pool of the genus Oryza. Next slide. And this is again the slide showing high level of resistance having the PI40 gene, and you can see the lines having that traditional varieties like Co39 and they are showing high susceptibility. So this gives the information that this gene can be utilized for blast disease resistance. Next slide. So here I want to make a point that we worked at IRRI to develop the, to identify the gene and help the national program. I am giving you an example. By doing 3 years research work, some of my collaborators in Turkey, they have a rice institute. They wanted to improve for blast disease resistance by using DNA marker assisted breeding methods, by using the gene specific marker for PI40 gene. And you see their crop was devastated by blast disease. But when they collaborated with me and introduced that PI40 gene, and the lines are showing very high level of resistance. So within 3 or 4 years time, next slide please. 3 or 4 years time, that program in Turkey, Rice Research Institute in Edirne in Turkey, they have developed 3 varieties having high level of resistance to this pericularia disease. That is only their target, how to develop pericularia resistance. You can see the line in the left side, and they have done marker assisted breeding, and they have released already 3 varieties to the farmers. So similarly in our country, also very soon, some new varieties will be developed from this exotic gene pool having very novel resistant genes. Next slide, please. Now coming to the third talk is about insect resistance. So far, there are several insect resistant genes that are identified from the conventional rice varieties or land races. I am talking about the gene from the exotic gene pool, which is Oryza australiensis derived gene, that is BPH18. So we evaluated that gene in many countries, and that is giving very high level of resistance to BPH population or biotypes in different countries. And that particular gene has been cloned in my lab, and then we are now using it in different countries for rice improvement program. Next slide, please. And this is the distribution of different genes for your information, how they are spreading from chromosome 1 to chromosome 12, but most of the time the clusters are present on the long arm of chromosome 12. Next slide. So my focus for presentation about insect resistance, about these 4 different genes. The upper 2 ones, BPH9 from Pokali, the land race in India, and BPH17, which encodes NBS LRR type of receptor protein. BPH17 encodes lectin receptor kinase protein for its defense against the insect. The BPH18 gene that I have cloned, it has 2 NBS motifs, NBA and NBS and LRR protein, and that defend against the attack of the BPH or brown plant hopper insect. The other gene is BPH32, which encodes short consensus repeat sequence domain containing protein, and that gene is derived from the land race Rathuhinati. So these 4 genes are very, very important for controlling the BPH insect attack. Next slide. So we have, for this purpose, for utilization in different countries, we have developed near isogenic line. So you introduce many clone genes into different in one genetic background so that scientists can utilize it, and whichever near isogenic line is showing resistance can be incorporated into their varieties. Next slide. Here, just you press it, it is showing the mechanism. Just press again, and it is showing, you see, how the insect is reacting to the protein, the defense protein, which has been cloned and characterized. One more, humans, one more. You see, insect actually it feeds from the base of the plant, then injecting and sucking the phloem sap from the plant. And then once that goes, so that receptor protein, which is NBS LRR protein, will be reacting to the insect. Either insect will go away or insect will die. Then your rice variety will be safe from the attack of the insect. Next slide, please. So here is the picture, what the level of resistance. You can see after screening that all the lines showing high level of resistance against the insect, and also both at the seedling stage and also in the adult plant stage. And you can confirm it by DNA analysis. Because of the presence or absence of the gene, in those lines, you are testing for the presence of the gene and its level of resistance. Next slide, please. So coming to that, what is the output of this? That how we are helping the national programs. So the scientists from NRRI and IIRR from Hyderabad, they have already incorporated these genes into our leading varieties like MTU 1010, Sorna, and Sambambasuri. And many lines are having blast resistant genes or pyramided genes for bacterial blight, blast, and BPH. So those lines are now under an advanced stage, and I hope that very soon they will be developing new resistant varieties for our country. Next slide, please. So some more information about the exotic species. I have given one slide that there are also some other species which has potential for a high level of resistance. In case there will be breakdown of resistance, other exotic gene sources, the scientists can work using the cutting edge science to clone the gene from other species which are also showing very high level of insect resistance. Next slide, please. So summarizing what I have explained, what I have presented on this particular topic, which is very relevant to the theme of this conference, that the exotic gene pool of Oryza opened a new paradigm towards rapid development of resistant rice cultivars by transferring biotic stress resistant genes towards producing sustainable yield of rice. Exotic novel resistant genes with specific defense functions, means the functions should be known. As we are in 21st century and we are using biotechnology, we should know the function and transfer it to different varieties, and those are identified and transferred into elite indica rice varieties. High value resistant genes, particularly I have listed XA21, XA27, XA40 for bacterial blight, PI9 and PI40, they are all from the exotic species, and BPH18 and BPH32 with the known functions are very ideal to improve indica rice varieties. Multiple resistant gene pyramids can be used and transferred to elite indica rice varieties that can confirm strong resistance to recently evolved biotic stresses of rice. Transfer of broad spectrum, bacterial blight, blast, and BPH or their gene pyramids into elite rice cultivars will provide durable disease and insect resistance. So with this, I conclude and thank you very much for your attention. Next slide, next slide.